Well, thank you so much for that kind introduction. It's a huge honor to be here. I'd like to thank the organizing committee for the opportunity to tell you some about our research. So the previous Symposium X were really fantastic topics, and it's an honor to get to and a little bit intimidating to follow those. But one of the things I'm going to try to do is incorporate some of what they talked about into my talk as well. So in my lab at MIT, we've spent the last several years trying to understand how biology makes materials. And one example of biology that making materials are shells. These are abalone shells and mussel shells. And these are made out of calcium carbonate. These are biocomposite materials. They're 98% by mass calcium carbonate, 2% by mass protein, yet they have very exquisite properties. They're 3,000 times tougher than their geologic counterpart. They're made in ocean temperatures and pressures. They don't use toxic materials, and they don't add toxic materials back into the environment. And so we think this is a great model for how to make future materials and technologies. And so I'm going to pass these around. I know you're eating, but you'll have the opportunity to look at them. These actually have pearls in them as well. Now, the geologic counterpart of these materials this is calcite here. This is a really beautiful piece of calcite, which I'm going to pass around. It is $200, so try not to drop it. It does have a perfect cleavage plane. And this is aragonite, the metastable crystal structure of calcium carbonate. So those shells are made out of both aragonite and calcite. And the proteins within those shells actually control the inorganic structure and the crystal structure of these materials. And so let me give you these different calcium carbonate structures to pass around. And so what we've been interested in the lab over the last several years is how biology makes such exquisite structures, and if we could get biology to work with the rest of the periodic table, because biology's done a fantastic job with shells, but not as much with materials like batteries and solar cells and other kinds of electronic devices. And so what our goal has been is to give genetic information to non-living materials so that they can have many of the same properties of biological systems, such as the ability to grow things at ocean temperatures and ocean pressures, environmentally friendly synthesis, and the ability to replicate. And so this is a collection of some of the faculty that I've worked with. I'll mention them as I go through. And I say that my students are my dream team because I've really had the most fantastic group of students over the last couple of years that have worked on this with me. And I have a little bit of a cold here. So we call it genetic control of materials. So the abalone, or the shells that are being passed around today, they have a DNA sequence which codes for the ability to make a shell, the calcium carbonate. Now, if you get a male and female abalone together, they make offspring, and they pass on to their offspring the ability to make this calcium carbonate-based material. Same with diatoms made out of silica. So what we said is, wouldn't it be great to have DNA information to be able to grow solar cells or batteries? And we think about future technology. We start at the beginning of the Earth, about 4.5 billion years ago. It only took about 1 billion years to have life on Earth, and then really fantastic things started happening. You started getting multicellularity, you started getting photosynthesis. But it wasn't until about 500 million years ago, during the Cambrian geologic time period, that organisms started making hard materials. Before that, they were soft and squishy materials. So what happened during that time? Well, there's increased calcium and increased silicon and iron in the environment, and now organisms had the opportunity to start making those materials. And so that's the Cambrian geologic time period, which the first company I founded is named after. And so we wanted to be able to have that ability as well. And instead of using complex organisms, we're going to use simple structures like viruses and bacteria, genetically engineer them to self-assemble and grow a structure like a battery. And so what we're interested in is the interface between nanoscience and biomaterials, because that shell that's being passed around, the tablets of those shells are nanostructured materials that are connected with the biological proteins. And by controlling the materials, the nanotechnology and the biomaterials, we think that we can have an impact in energy, healthcare, and the environment. And the secret really is the control of the materials at the nanoscale. It's the same for the shells, but it's also very important for materials in solar cells or batteries. And this is basically the fields that we work in. We work in solar cells, energy storage, fuel, enhanced oil recovery, CO2 sequestration and storage, more abundant materials, water purification, therapeutics, and imaging. And I'm going to hit on a couple of these topics today. And we have partners, companies that we work with, companies that I founded, as well as Mass General Hospital. And so here's some of the natural biomaterials. This is a coccolithophore that's a unicellular algae made out of calcium carbonate. This is the shell that's being passed around. And if you look at SEM image of it, you'll see that it's made out of these stack of coins of aragonite, that metastable crystal structure of calcium carbonate. The diatoms are made out of silica, and there's iron oxide used in magnetotactic bacteria. And so we think life has done a great job, but we think life could have done better. If you go and you look in a scanning electron micrograph, you'll see these individual tablets, and in between each of these individual tablets are a protein that are coded by the DNA that actually control the crystal structure and the stacking of this calcium carbonate-based material. And so what we decided to do is see if we could develop a protein interaction between any inorganic material we wanted so that we could start to grow that material or bind that material and organize it in a way using biological self-assembly. And so if you look at what nature's done so far, it's done a great job with calcium in the form of calcium carbonate, silicon in the form of silica, iron oxide, Fe2O3, Fe3O4, but really has left a lot of the periodic table unused. And so we think that if the ocean had the ability to process these materials, organisms had the ability to interact with these ions, maybe we could have materials that were new materials that biology hasn't had the opportunity to work with. And so instead of using complex organisms, we use M13 bacteriophage, which is a virus that infects bacteria. To material scientists, it's an object that's 880 nanometers in this direction and 6 nanometers in this direction and has single-stranded DNA. So what we're going to look for is a DNA sequence that codes for a protein that can grab onto an element or a material and be able to bind it and organize it and assemble it into a device. And so using this single-stranded DNA, we go in and we put a random DNA insert in here that codes for a random protein insert on this end of the virus. And if you do that 1 billion times, you can build a library. So we build a library of information. So this is the DNA. We go in and we put a random DNA insert that codes for a random peptide insert and express that millions and billions of times. And now you can have a library. So this is called a phage library. So each of these viruses are going to be genetically identical, but they're going to differ from each other based on a small peptide insert, a short protein. So we can take a library of 1 billion different possibilities, force it to interact with any material we want, and what we're looking for is trying to replicate that kind of interaction that is in the abalone shell, a protein that will recognize calcium carbonate. Because the proteins in the abalone shell are very negatively charged. They grab ions out of the environment, and they can put down calcium carbonate, and start building a structure. So we want to be able to do that with any material we're interested in. And so we're basically trying to make an imprint of that material. You throw 1 billion possibilities at this material, most of them won't bind. You wash away the ones that don't bind. You take the survivors, the kind of a Darwinian process, the survival of the fittest, and the viruses can't replicate themselves, so you infect them into a bacterial host and make 1 million copies. So what you do is you have a rich population of a virus that recognizes some material. Now you ask it to bind again under more chemical stringent conditions, and you look for the 1 out of 1 billion that will grow a better battery. And so my students made this cartoon. This is my very good collaborator, Professor Paula Hammond, who I think spoke here yesterday. And our idea is to give genetic information to all these structures that normally don't have genetic information. Batteries, new kinds of catalysts, drop-in fuel replacement. We have a program where we genetically engineer yeast to take waste CO2 and build tiles for green buildings. We have a program, which I'll talk about towards the end today, where we've developed virus-based inorganic materials to find tiny ovarian tumors. We also work on photocatalytic splitting of water and other building nano antennas using viruses and chromophores. And so all of this is the idea of giving DNA where DNA normally didn't play a part in these material synthesis. So here's the M13 bacteriophage. It's 880 nanometers in this direction. It has DNA sequences, or it has a DNA genome that codes for proteins. So on this end, there's protein 3 and protein 6. The major coat protein, of which there's 3,000 copies, covers the DNA. And on this end, there's protein 7 and protein 9. We can go in and individually modify the DNA to individually modify the proteins to be able to express proteins of interest or random proteins to look for the best one for a particular application. Now, the M13 bacteriophage is actually quite a beautiful structure. It's made out of almost 3,000 proteins that are alpha helical. And these alpha helixes actually self-assemble around the DNA, and it forms a crystalline structure. So what we want to do is actually keep this structure of the M13 bacteriophage and display on it peptides of interest so that we can use these as nucleation points to start growing and assembling materials. And everything we want to do, we want to try to be able to do it under room temperature conditions. Now, if you go and look at these alpha helixes, what they really are is this beautiful assembly of amino acids with specific functional groups that can be thought of as kind of a reaction vessel where the synthesis can take place. The synthesis or binding of the materials can take place. And because these structures are long and thin, we started thinking of them as wires, where now they'd have the ability to start grabbing ions out of solutions similar to an abalone shell and start to build either polycrystalline materials or single crystalline wires. And we were really excited because we were able to build single crystal wires out of these materials. But you can see in this TEM image, this is a 200 nanometer scale bar, and so we couldn't make very many. So we spent many years trying to learn how to make a much higher concentration of these so they could be used for applications like solar cells and batteries. So here's kind of an example. This is one phage that we call P8 number 9. It was a phage that was first selected in my group from Chung-Yi Chang, and then Yujin Lee followed it up to be able to grow materials for catalysis applications. And so in this particular case, we have this protein we selected against gold. It's really good at taking gold ions and building them into gold nanowires. And so this is a test tube of 3 different clones that we tried. This is our clone of interest. It's called P8 number 9. If you put it in solution with gold, it's able to bind the gold and make nanowires out of it. But some of the other solutions that you use actually have no affinity for the gold or very little affinity for the gold. So you can easily find a sequence that's specific for a kind of material. Now, you can use these as templates to grow gold nanowires of different thicknesses and eventually different lengths. You can use it to grow different alloys of materials. This is a gold and silver alloy, so different concentrations of these materials incorporated into the virus nanowires. And then you can do other fun things like have it first grow gold and then grow platinum on top of it to make core shell materials. And this was used for fuel cell applications. So just going through the periodic table and kind of materials, we've been able to use these virus templates to be able to grow and assemble many different kinds of materials for applications for energy storage, catalysis, magnetic materials, energy storage, solar, and other solar applications like photocatalysis. And the other thing that's great about this is that you can go in and make other modifications. You can do hierarchical assembly. You can add other kinds of functional groups to it. We've evolved these viruses to function in other solvents. And we've put synthetic active sites in them as well to have organic-inorganic hybrid materials. You can think about making high-order assemblies of these. You can make hydrogels through molecular interactions of these phage virus complexes. And you can use them to make copper and nickel materials, different kinds of nanofoams. This is copper and nickel foams that are used for battery applications. So one of the things that we really wanted to do was mix different kinds of materials at the nanoscale. And so in this particular case, we're thinking about this phage as a mediator to bind 2 different materials. And so one material may be a carbon nanotube, and the other material may be an inorganic nanoparticle. Now, normally you can think about mixing these through chemical mixing, or you can think about mixing them through so physical mixing or chemical surface modification to be able to bind them. But sometimes on the nanoscale, if you change the chemistry of the surface, you're going to change the function of the materials or the property of the materials. So we decided to use a virus as a mediator to first bind the carbon nanotubes and then bind the titania through having 2 different kinds of affinity molecular interaction. And ultimately, what we want to do is be able to go from a DNA sequence to a functioning device and be able to have a sequence that codes for a device. And so one of the first kinds of applications we looked at were lithium-ion batteries. And we first looked at the ability to grow anode materials and then to be able to grow cathode materials. And so for the anode materials, what we decided to look at was be able to find a virus that could grow cobalt oxide nanoparticles with high affinity and very nice small cobalt oxide nanoparticles. Then we went in and did additional genetic engineering to give it a second gene because we wanted to be able to find a virus that could make a material that was a good ionic conductor and a good electrical conductor at the same time. Then we use the kind of power of biology to self-assemble these materials and make it into a functional battery. And basically, you make it just in at the bench where you have your virus sequence. You give it a cobalt source, and you're able to nucleate these cobalt oxide nanoparticles, and then we can test it as a battery electrode. So this is specific capacity versus cycle. This is commercially available. This is theoretical capacity, and this is what our first virus battery looked like. I always say this is the first data my students showed me. And so you can see that it hits theoretical capacity at first, and then as you cycle, it starts to fade. And so what my student, Kite, decided to do was go in and give this virus a second gene, one to grow gold and another one to be able to simultaneously grow cobalt oxide. And so this is a virus that's binding gold. Now you give it cobalt oxide and it zips up the structure. And now you have a cobalt oxide gold complex, and you can test that as a battery electrode. So this is capacity versus cycle again. This is the first generation battery. This is second generation battery. So through engineering and selection, we're able to increase the performance of this battery. So we decided to look at cathode materials, and what we decided to do was take a virus to see if we could get it to bind a single-walled carbon nanotube on its tip, and then simultaneously have the ability to grow an iron phosphate material, and then wire itself to the current collector. And these were two very talented students, Yoongjung and Hyungjung, in my group. And they were able to accomplish that. They were able to select viruses that could bind single-walled carbon nanotubes on the tip and then have a different sequence that could nucleate iron phosphate material along the edge. And this is a TEM image of what it looks like. They can form a percolating network of these virus CNT complexes that are now mineralized with the cathode material. And basically what happens is it forms a superhighway to get electrons through this percolating network and transfer energy in a very short period of time. And what we found was that this is specific energy now versus specific capacity. This is a 1-gene battery. This is a 2-gene battery, and this is our best 2-gene battery. It has the best DNA sequences that bind the carbon nanotubes the tightest and are very effective at growing the iron oxide, the iron phosphate material. And so basically we were able to get a very good battery that was a kind of record-breaking battery at the time, and that battery went to the White House for a press conference that President Obama was giving on clean energy, and this is the virus-based battery right here. So this was our first virus-based cathode material. We've been able to make lots of other ones of various different kinds of material. This is one I saw Miriam in the audience made that we like to show that you can use for useful things like showing off MIT. And this was President Obama visiting Professor Hammond and I to see the battery at MIT. Now, we showed that you can bind carbon nanotubes. How about graphene? So that's trying to incorporate the second BioX talk into mine, which is we thought about taking viruses that could bind graphene and give them a second sequence that would simultaneously allow them to nucleate a material. And so this was bismuth oxyfluoride material. And so what we found is that we could find viruses that could bind to this material really well and then nucleate the material of interest. When we showed a great enhancement in performance with those kinds of materials. And so then we also decided to look at lithium-air batteries. So the first part was on lithium-ion batteries. We thought, well, is there anything a virus can do for lithium-air batteries? Well, these are very promising materials for long-range vehicle applications, but there's a lot of current limitations, including a large overpotential, a low current density, short cycle life, and cost efficiency. And so we said, well, what can we do to see if biology can make an improvement here? And so this is work from Dahyun Oh from my group. And what she did was she genetically engineered viruses to grow materials for the catalyst electrode. And the idea is one of the problems with cyclability is that the discharge product of lithium peroxide builds up on this electrode. And so we wanted to figure out a way to both increase the catalytic efficiency as well as decrease the ability of the TiO2 to block holes and pores so that we could actually increase the ability to cycle this material. And this is a movie that she made where basically you have a virus it's in water. It has the sequences to be able to grow a manganese oxide material. And you give it a manganese ion source, and you grow these manganese oxide, these very spiky, high surface area materials. Now we give it a second ability to be able to bind palladium, and then you can test it as a electrode material, lithium plus oxygen plus 2 electrons from lithium peroxide, and it's a reversible reaction. Now you can take a lot of those and get them to self-assemble and test it as a lithium-oxygen battery in a system that would look something like this, where they self-assemble. And we showed that it was functioning, and at the time that we published it last year, was the best, had the best performance of a manganese oxide lithium-oxygen battery. And so here's my car. And no, it's not really, but what you would be able to greatly increase the distance between charges. And so why is this? Well, one of the reasons is when you get these viruses to form in these wires, they also have these spiky materials, the spiky manganese oxide material, and it forms this porous network, and you have increased surface area. And so we think it makes it easier to have the manganese oxide and the palladium surface interact with the ions in solution and allow for the reversibility of the lithium peroxide. So what we did was take the virus, grow a manganese oxide material, wrap it with PAA that would bind to the metal nanoparticles in solution, and we could do that and actually further increase the performance of the battery. And so this is a manganese oxide nanoparticles. This is biotemplated manganese oxide nanowires. And this is biotemplated manganese oxide with the palladium. And you can see that we get a 45% improvement of specific capacity when you have this whole complex together. And you have a 7 times higher current density. And it only has a small amount of the noble metal, 1 to 3% for this performance. So we were pretty comfortable working with virus-based CNTs, and so we said, well, let's see if we can use those for dye-sensitized solar cell applications to try to increase electron collection in those materials. And so basically in a dye-sensitized solar cell, you have a dye molecule, you shine light, you promote an electron. That electron gets injected into the titania particles and has to make its way through this titania network to the current collector. And so we said, well, what if we put a virus in there that could help get the electrons directly to the current collector? And so we engineered this virus to now pick up single-wall carbon nanotubes and line them around the surface of the virus. And so you can put 1 carbon nanotube on a virus, or you can put up to 10 carbon nanotubes on the virus, and they're each held in place by about 200 peptides that are specific to bind these carbon nanotubes. And these were found by taking a library of these bacteriophages and interacting them with carbon nanotube film and finding the one that would bind the best. Now, what we wanted to do was be able to reduce the surfactant and use the virus as a surfactant. We were able to find virus sequences that could act as a surfactant. So what you're looking at here is dialysis, where we're removing the surfactant and replacing it with different clones of these viruses, and you can find a few viruses that will basically stabilize the CNTs in solution where you don't have clumping. And one of the major things that we were excited about was that it had histidine in it, which was going to be useful for controlling the synthesis and the reaction. So what we had viruses that could pick up carbon nanotubes around their lengths, and then we could have a sequence that could nucleate titania out of solution. So now we had titania grown over our virus-based carbon nanotubes, and this was anatase. And so what we could do is now take our virus complex, grow the nanoparticles, and then put a small amount of these virus carbon nanotube titania complex into an already existing paste of titania and see how it would increase the efficiency of the solar cell, or if it would. And so this is actually what our solar efficiency for these dye-sensitized solar cells without CNTs. we were able to get an increase of 33% by adding the CNTs in just a couple of percent incorporation, where they would provide a path for the electrons directly to the current collector. And we compared these to ones that either had surfactant or as produced CNTs. So the as produced CNTs would actually clump in the structure without the virus, and it would decrease the efficiency of the solar cell. Another area we've looked at in energy is people have asked me to work in biofuels for many years. And I was never really comfortable in figuring out a role for viruses in biofuels. But what we thought about was, well, we can get DNA sequences that can control proteins that can control calcium carbonate. What if instead of using a bacteria or a virus to produce directly produce your fuel, what if you made new inorganic materials, new catalysts, and you could then take those new catalysts and be able to use them for fuel production? And so this is the company that I founded, Siluria Technologies. And our idea was to work on a low-temperature oxidative coupling of methane. So what we want to do is to produce ethylene. And normally, for the production of plastics, you'll have oil and or a longer chain hydrocarbon, and you'll break it down. What we wanted to do was take methane and build it up one carbon at a time. So basically, we wanted to take the structure of methane and find a new catalyst for forming ethylene. And we wanted to be able to do it at low temperature. We wanted to be able to do it at reduced carbon footprint and economically feasible. And so we had a great team of scientists that came on board and worked on this. It's a very difficult process. People have been trying to work on how to do it economically, low temperature, about 30 years, and hadn't been successful. And so we said, well, what if we let biology work on it and see if biology can help solve the problem? And so what we did at Siluria was take combinatorial chemistry, combinatorial biology, and rapid throughput screening to see if we could find new catalysts for this reaction. And we were able to find nanowire catalysts that were made of a combination of different elements, and we were able to find about 10 catalysts that were effective in this reaction, and with a 60% lower carbon footprint. And so we now have several locations. This is headquartered in San Francisco. We have several pilot plants. We have a demonstration plant for this reaction, which is making ethylene, and from ethylene going directly into gasoline and diesel. And in about 2 months, it should be on board in La Porte, Texas, for making a ton of ethylene a day. And so this process was very difficult. It took out of the box thinking, a different way of thinking about how to design catalysts that used a combination of combinatorial biology and combinatorial chemistry. Another company that I founded that is inspired by biology is Cambrios Technology. So both of them are named after geologic time periods. And the idea here was to be able to use nanomaterials for touchscreens and for transparent conductors. And what we wanted to do was replace indium tin oxide. And inspired by biology, we wanted to do wet chemistry. We wanted to be able to do low-temperature synthesis and processing, and we wanted to be able to do it in a way that was environmentally friendly, not using toxic materials or adding toxic materials back into the environment. And so at Cambrios, we developed a technology called Clearome, which is a silver nanowire technology. The inspiration came from bacteriophage, and some of the chemistry to work out how to do this grow these nanowires came from biology, but there's no biology in this final product. But we can make very flexible materials that can be made on very different substrates. It's highly conductive, extremely transparent, flexible, and formable. It's processed at low temperatures, and now it's available in large volumes and used for touchscreen devices. This is one of the Cambrios phones that's now on the market that I have here. But we have several products now on the market, and about 5 more coming out at the beginning of the year that are monitors and all-in-ones, smartphones, and tablets and e-readers. So again, we were able to overcome this problem of difficult to process, less environmentally friendly or less environmentally abundant materials by thinking about how biology would approach the problem. And now we've been able to deliver on that and have products that you can buy The last story I want to tell you about is about cancer. I'm in the Cancer Institute at MIT, and a couple of years ago, Professor Bob Langer asked me to come work on cancer, and I was a little bit intimidated about it because it's such an important problem. And so I said, well, let me see what we can think about in terms of making new materials that might be important or may have an impact in cancer. And this is a collaboration that we have between MIT, Professor Sangeeta Bhatia at MIT, and my really fantastic graduate students and postdocs, MIT's Lincoln Labs, and MGH. And what we wanted to do was develop a way to see tiny tumors and develop a surgical guided system to be able to remove small tumors during surgery, and eventually develop a system to be able to diagnose small tumors in a less invasive way. So we started thinking about optical imaging, and we decided to look in what's called the second window near IR, which is imaging above 1,000 nanometers. At this wavelength, you can have penetration depths that are up to 10 centimeters. At least that's what the theory says. It's non-radioactive, it's inexpensive, and it's quick imaging, but it requires sensitive equipment for detection. But our whole imaging system costs about $200,000 right now. We're building one for about $100,000 compared to the millions of dollars it costs for an MRI. And so we thought, well, maybe we can come up with a way of looking deeply into the body in a way that would be portable, that could be used in underdeveloped countries where MRIs may not be possible. And it also could be used directly in a physician's office. And so imaging in this second window near IR is very interesting because biological objects are much more transparent than they are in first window near IR. And as I said before, it's thought that they could go up to about 10 centimeters in depth. You also have less autofluorescence there and improved signal to noise. And so we developed a home imager in our group that was based on an indium gallium arsenide detector, an 808 laser, a system where you can image mice, phantoms, or you can do cell cultures. And initially, we were donated an old animal imager and guided it, and my students, Jifa and Rana and Hyung Jung, and now Shengnan and Neil, built these first couple of generations of instruments from scratch. So the problem we decided to focus on was ovarian cancer. About 250,000 women are diagnosed yearly worldwide, and about 75% of these patients present with advanced stage disease. It's difficult to detect this disease early. About 80% of them respond with chemotherapy, fortunately, but a good number of those patients actually develop a resistance to the chemotherapy. And so basically, you have a minimal increase in survival rate over the last 30 years, and there's about 140,000 deaths per year. And so it's obviously a very important problem. Right now, the gold standard for diagnostics of this is contrast-enhanced CT, and it has poor sensitivity at small sizes below a centimeter. And so we decided to see what's the smallest size that we could locate using our imaging system. So how do we look at it? Well this is actually, the treatment for this right now is surgical debulking. So a surgeon goes in and by eye removes as many tumors as he or she finds, and then it's followed up by chemotherapy. And the outcome of the patient is very reliant on the optimal debulking, or the optimal removing of these tumors. And so if your surgeon's able to find most of the tumors and remove them, you greatly increase your survival rate. And so what we wanted to do was build a tool to be able to help the surgeon find the small tumors. And of course, we use a N13 bacteriophage. And so the system that we decided to use was to be able to find a phage or a virus that could be able to target ovarian tumors on this end and carry with it single-walled carbon nanotubes, which we knew we were really good at being able to bind and place where we wanted them to go. So we used an expression for ovarian tumors called SPARC. It's a peptide that's overexpressed in ovarian tumors. So we were going to be able to deliver this we started in mice, IP, basically in the interperineal space, to a mouse that had ovarian cancer and try to target our probe there and then image it. And so this is actually showing how little background there is in the second window near IR. So this is pre-injection. What we're imaging is a mouse that has a high tumor burden. And this one is pre-surgery. So all these spots you see are a collection of tumors, ovarian tumors, in this particular mouse. And so we had the surgeon either have a map of this to begin with or go in without our map. In this particular case, he went in without our map, and he removed all the tumors he could see by eye. And then he stitched the mouse back up, and all the fluorescence you have left behind are the tumors that he didn't see by eye. Then we gave him a map of where we thought the tumors are, and he went in and removed many more of these tumors. And so it basically greatly increased the small tumors that he could see. Not only that, there were tumors that might have been larger, but he couldn't see because they were behind other organs or were just not visible. And so our map gave him the ability to find tumors that would be difficult to see. And so in this system, basically, this is looking at the nodule diameter of the tumor cells with SWNT-guided probe or unguided. And he's a very good surgeon, and he could find small tumors by eye. But when it got below a millimeter or below 2 millimeters, it got difficult for him to be able to find them. And so in this case, he was able to find 30 tumors that were below 1 millimeter below in size, and it was able to pull them out. We're doing survival studies on these right now to see if he was able to increase the lifetime of these mice due to having a higher removal of the tumors. Now, the next generation instrument that we're developing, that we've developed, is a real-time surgical imaging system where we can inject the probe into the animal, and the surgeon can see in real time where the tumors are, and he can monitor it as he removes those. So this is my group, members of my group that contributed to this, and collaborators that I tried to mention as we went along. It's a fantastic group of students and postdocs and collaborators that have been really fantastic to work with over the last couple of years. Here's some of the funding agencies that have funded this work on virus-based growth of materials, as well as some of the initial funders of my companies. So I know that was pretty fast. I was trying to cover batteries, solar cells, and cancer. And if you have any questions, I'd be happy to take them.